All right. General error starting recording. Okay, I'm going to go ahead and start the broadcast.
The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon. Welcome to the CFIDS Association of America's 2010 webinar series. I believe this is the 8th program in our 2010 series, and we're delighted today to provide some information and some exciting news about the SolveCFS BioBank. My name is Kim McCleary. I'm President and CEO of the CFIDS Association of America, and we have two additional speakers here with us this afternoon who I will introduce in a moment. And if we could take just a couple minutes, I'll orient folks. We have about 200 people who've registered for the program this afternoon, and I think it was about 40% of them indicated that this was their first webinar. So I'll just go over some quick kind of housekeeping details for those of you who are new or who haven't participated before in one of our webinars, and then we'll get right on to it. First of all, just to let you know, there are 3 people whose voices you'll hear this afternoon, and we are all 3 in different locations, kind of spread around the country, and we can't see one another. We can't see you, and you can't see us, which is definitely a downside of this technology, but the upside is that none of us had to go anywhere today, including all the participants who are joining us online. So it's a great way to deliver programming, but none of us have the benefit of being able to get body language and informal communication cues either from one another or from our audience today. So bear with us with this somewhat isolated community experience. It's the best way to describe it, maybe. As people registered for today's program, you were able to submit some questions and also to respond to, I think, 3 questions that we put out. Those answers that we had received by last night were shared with Dr. Horn and Dr. Vernon in advance. And you also have chance to submit questions during the presentation that hopefully we'll have time to get to before we wrap up this afternoon. So go ahead and type those questions as you think of them. They all come into me, and I'll do my best to kind of triage things that are of a more technical nature, questions about where to find different links and things during the presentation, and then the 3 of us will just take your questions that have come in at the end. We will be recording today's webinar if the technology cooperates, and we'll have the recording and the slide decks posted on our website. We've been posting the webinar recordings on YouTube because that seems to be a more accessible and user-friendly format than on our website, but you can find links to all of that on our website, and the URL for that will be delivered in the follow-up email that you receive either tomorrow or Thursday, depending how quickly we can get that posted. Just one reminder, both because both of the speakers are PhD doctors and not MD doctors, we're not able to address individuals' questions about symptoms or treatment, and also this format does not lend itself. Even when we do have a physician speaker, it's a difficult way to address questions of a more personal nature. So we won't be able to take those types of questions. But otherwise, we welcome you to type into your screen, and if you just take a moment to look at the top right-hand side of your computer screen, you'll see a little dashboard there with a question box where you can type questions, and those come through the technology and the electricity to us. Also, if you're having difficulty with either seeing things or hearing things, let me know that too, and we'll try to make adjustments. Somebody's already let me know there's an echo in here, so we'll try to take care of that. And each of us are on a different system, and it might be better for one and worse for the other. So we'll try to handle that as we can. So take a second now, if I can get my slides to work, to introduce the first of our speakers, and it's my pleasure to have Liz Horn, PhD and MBI, with us here this afternoon. Liz is the Genetic Alliance BioBank Director. She holds her doctorate degree in molecular pharmacology and has worked in the nonprofit sector for, I believe it's been 7 years. She's the former director of research at the National Psoriasis Foundation and has had a lot of experience with psoriasis and setting up biobanking with a couple of different organizations. She also serves on a number of other biobank groups, including the Cancer Human Biobank and the NIH Biospecimens Interest Group, which is, I think, more informally known as BIG. So Liz brings with her a wealth of experience to Genetic Alliance and has been just a tremendous partner in helping us get the SolveCFS BioBank set up over the past, gosh, it's really only been about 7 months, I think. We've had a really rapid start, and it's a great deal of that is due to Liz's tremendous help in getting us to this point and having today's program as a sort of foundation for explaining what the biobank is and how biobanks more in general are used to facilitate and accelerate research. We're also going to hear from Dr. Suzanne Vernon, my colleague at the CFIDS Association of America. Suzanne is our scientific director and has been with us since the end of 2007. She holds her doctorate in virology and also has a certificate in public health genomics, which is how we came to learn about Genetic Alliance through a certificate program that Suzanne took part in. And as you've heard from Suzanne, if you've participated in other of our webinars on the association's research program, Suzanne is very much pursuing a systems biology approach to CFS. You'll hear from both Liz and Suzanne in just a few moments, but before we do that, I wanted to just introduce a couple of other people who are on the line but are too shy to speak today, who are involved in our biobanking efforts. And for those participants who are already in the enrollment process for our biobank, you've probably talked to Gloria Smith, our biobank coordinator, who's responsible for handling all of the consenting and information delivery and keeping in touch with our biobank enrollees as they step through the process. And we have just added a college student, Lauren Kenny, to help us with the biobank data that we're accumulating through the patient questionnaires. And Lauren is a biology student at Agnes Scott College and is using some experience she's had in helping convert medical records to electronic format with the biobank. So we're excited to have just a real great team of people leading us on the biobank. So I'm going to switch back to Liz's slide and hand the controls over to her and ask her to lead us through how Genetic Alliance came to be in the biobanking infrastructure business and how your organization facilitates organizations like ours to be involved in this process. Liz?
Thank you, Kim. Can you see my slides?
Yes.
Great. Thank you so much for giving me the opportunity to speak with you today. My name is Liz Horn. I'm the director of Genetic Alliance BioBank. I've been involved in advocacy-initiated biobanks since about 2004 when I joined the National Psoriasis Foundation and set a biobank up for them. And one of the things that I really want to tell you today is about the important role of advocacy organizations in biobanking. I think this is a a new field for advocacy organizations, but it's a really, really important one because really we're all better suited to do this in many ways than traditional researchers and clinicians. So just to begin, what is a biobank? And
Liz, could you put your presentation into slide mode so you can take up the full screen?
PowerPoint will close.
Oh, I'm sorry. Okay.
Yeah, sorry about that. I'm not sure why I have that problem.
Get to see that you have a really cool Mac.
Yes, I am on a Mac, so that is probably why. So what is a biobank? A biobank is a research resource or archive repository in which a major component is biological samples, and they can be taken from different individuals. They can even be taken from different species. I would like to add that I think data is an incredible aspect of the biobank, so it's biological samples as well as data. And Genetic Alliance BioBank is a group that provides a shared infrastructure for disease advocacy organizations to manage both biological samples and clinical information. And what's happening now is the landscape of biobanks is really, really changing. They used to be very much single condition, single siloed collections, maybe controlled by one or two people. There probably wasn't as much information, and really participants weren't very active in them. And what we're seeing now is there are many multi-condition cross-disease biobanks. There are now models for shared infrastructure like Genetic Alliance. Groups are governing them. Disease advocacy groups are playing a huge, huge role. And we're coming to a lot of increased standardized protocols for samples and data, which will help the science. And we're getting data from multiple sources. And one of the major changes is really the dynamic involvement of the consumers and the participants. And the Advocacy BioBank is a wonderful, wonderful platform for this because you can bring all the stakeholders together in a way that the other stakeholders can't really do. So you can bring together the medical community, industry, the researchers, and the patient community as well. So how did advocacy groups get into this? And this really, this is sort of the worst case scenario, and this happened a lot of times in rare diseases in these much smaller communities. But this was really the impetus of why we formed Genetic Alliance BioBank, that maybe there was insufficient informed consent. There were small collections that didn't have enough power because they were too small, and you would have the same samples from the same people in researcher A's lab and in researcher B's lab. Phenotyping was variable. The legal structures didn't protect anyone, and you ended up with these collections that were started but were never followed through with. And the participants were really frustrated because they had given their samples, and the scientists were frustrated because they didn't have what they needed to do their work. So why should advocacy organizations get into this space? Well, they really understand the unmet research needs of the community. They're able to develop trust within the patient community, and it's a testament that there are 200 people right now on this webinar to that. They can leverage scarce resources. They can be very efficient. They can share costs, and they can facilitate collaboration between stakeholders, between clinicians, scientists, industry, and sometimes individuals that it's very hard to get them to the table together, but they can all come to the advocates table. They're very committed to the cause. They're the best stewards of the collection, and their main goal is to make sure that the collection is sustainable and that it exists while continued research is needed. And again, they can bring many people together at the table. And so while this is a new space for advocacy to participate in biobanks, there are a lot of advocacy organizations who are doing this. We surveyed 124 organizations through Genetic Alliance, and almost half of them were involved with a registry or biobank. And this is in addition to other traditional advocacy roles like developing materials, publishing a newsletter, organizing support groups, and about half had a support line. So how are these advocacy organizations involved? Well, 50% of them, they've established a registry or biobank. They've provided financial support. They help obtain financial support. They recruit individuals. They require researcher permission to receive samples, and about 40% own the registry or biobank. And they're collecting a variety of things: names, medical information, medical history, and a variety of samples, blood or other biological materials. So CFIDS is very, very well poised, the CFIDS Association, to be part of this community. And there really is impact on research. Data quality increases, the financial costs go down, there's greater participation rate, and the people that are participating are much more engaged. This results in a shorter time for research to be completed because it doesn't take as long of a time to get the samples. The participant risk can be minimized, and it really helps individuals make more informed disease decisions and increasing the amount of disease research. And that's ultimately very, very important. So to do this, in 2003, we formed Genetic Alliance BioBank because we realized that advocacy groups really should be leading these kinds of initiatives, but not all advocacy groups have the infrastructure to do this, and plus it can be expensive. So this is the group that, this is our advisory board. We founded it in 2003, and our vision was to revolutionize access to information and resources. And we did this by providing registry and biorepository solutions. And we did this in a cooperative model. So it was an extensible, interoperative, cost-sharing model. So if you think of other co-ops where you buy into it and then you're all sharing the cost, so really you're getting a discount on the services. So it makes it much more cost-effective. And what we've done is we've really built this infrastructure to train and mentor the advocacy organizations who are coming into this to manage and steward their own collections. We collect clinical information as well as biological samples. Everything is web-based. Everything is customized for specific disease and condition needs because everyone needs to collect specialized information. And so we can do all of that. We provide a lot of training for the people who are running the registry and the biobanks through different training sessions, manuals, webinars. And we've really built a robust system that can be accessed from anywhere, which is so important because in today's world, so many people are in different places, and especially in advocacy organizations. And it's a testament today that we're on both coasts here on this webinar. So how does this work? So it's a cooperative research consortium. There's an advisory board that represents the organizational members, and we work to configure the best possible infrastructure and continually updating things as the science and technology improve, but also as the needs of our member organization changes. So as these needs change, these groups can say, oh, we now need to collect this, and we figure out how to do it. And our members bring passion, skills, and resources to the collective. They leverage collective resources, and as we have more members, we have the ability to have more services. And we enable cross-disease research. But each member group, they govern and steward their own collection, so they maintain the control of their collection, which I think is really, really important. We're just here to help them do it, but really it is their collection. So the CFIDS Association BioBank is CFIDS Association BioBank. So here are our member organizations, and you can see Suzanne, CFIDS Association of America. And while we started out with some smaller rare genetic diseases like PXE, like Joubert syndrome, you can see that we have different conditions. Psoriasis is a chronic disease. It's a common disease. We also have a breast cancer group and hereditary angioedema. So we have a diverse group of member organizations. They're all different sizes of organizations. Some have staff, some don't, some are all volunteer. And they're all able to use this infrastructure to create really robust collections for their communities. So how does this work? This is a schematic I put together of the sample collection and data process. The member organization, so in this case it would be the CFIDS Association and the individuals working on the SolveCFS BioBank, like Gloria, they would consent eligible participants. And the participant would provide their name and contact information. And then the member organization would then enter that and request a kit from the biorepository. So we have ways to communicate with our biorepository that's located in Gaithersburg, Maryland. And some of you may have already experienced this process. The kit is then sent to the donor's home, and the donor then has the blood drawn and a buccal cheek swab collected. And the sample is then sent back to the repository for processing and storage. And ultimately, the sample will be sent to researchers. A very similar process happens for data. And after the participant is consented, the member organization will request a clinical questionnaire, and that will be sent to the participant by email. The donor will complete the questionnaire, and the data will be deposited back into the registry, and that data can then be made available to researchers. We have a variety of vendors. Our biorepository and registry are at GeneLogic. We are also working with Rutgers University to process and develop cell lines. And we have some other vendors like Exam1, which we use for phlebotomy services at specific events. We work with the National Center for Biotechnology Information. I can't remember exactly what NCBI stands for at the moment. And we also work with Private Access, a company that is providing dynamic online consenting, which we are rolling out to the biobank. So we've developed this customized biobank solution where there are sample collection services, clinical data management and analysis, and sample processing. And all of this can be accessed online through the web, and it allows our member organizations to administer their collections from wherever they are located. And they also have the ability to manage their inventory, request kits to be sent to donors, request samples to be sent to researchers. And we have multiple different systems, and most people who are in the biobank won't see all of them. There's a back-end registry called TRIMS. There's an action request portal where the member organizations communicate with the biorepository personnel. They ask them to send kits. There's an online questionnaire data collection system, which is what most participants will probably see the most when they're filling out their questionnaires. There's both the user interface as well as an administrative interface for the member organizations to use. We also have a data mining software that will help researchers query and visualize anything that's in the database. So they'll really be able to mine the data and ask some complex questions to help us really understand more what's going on with these conditions in our biobank. And then all of these systems work with each other. And so really, disease advocacy organizations are completely changing the landscape of biobanking, and it can't be done without their dedicated community and the involvement of the people in their community like you. And so what have we learned? And so we've been at this for a while, and I've been working on this both as someone setting up an individual biobank and then helping many other groups set up biobanks, both through us and those that have done other efforts on their own. We really don't want to duplicate efforts. Registries and biobanks are expensive and they're administratively burdensome. They aren't easy, and so if we can work together, that's critical. When it's possible, we really need to develop partnerships to share data and resources because resources are so limited. And we need to ensure that the biobank is sustainable over time. That's really critical. And one of the things we do with our member organizations is we work with them to make sure that they have the capacity to engage in a research project like this. And again, this the CFIDS Association of America is really, really well poised to do this. But some of our much smaller groups we really work with them to make sure when they want to start this that they really are ready. If you can use common data fields and controlled vocabulary, that will allow comparison across multiple data sets, and we've implemented that, and we're really working closely with Suzanne on your questionnaire to make that happen. Retention is key. It's so much harder to recruit new participants than it is to keep those that are already participating. So really having an active community engaged in this is really what's going to set it apart from other similar initiatives. So the engaged community and active participation is really critical. And there's a lot of planning that goes into this. I mean, I've been working closely with Suzanne and Kim for months now, and they've really done a fantastic job, and they're off to a very, very good start, and they've done this so much quicker than some of our other groups have been able to do it. But they've really been working hard over many, many months to get to this point. And I'm coming to the end of my time, and I just wanted to share a quote that one of our participants shared with me actually by email when I was with psoriasis, and I thought it was really eloquent that seldom is it that ordinary people like myself get to participate at any level in extraordinary events. I see donating blood to the biobank as one of those events. And now I would like to turn it, I think I'm turning it back to Kim, or am I turning it over to Suzanne?
You can turn it over to Suzanne directly.
Okay, so I'm going to change presenter.
That was such an outstanding quote from that particular participant. I love that.
Oh, and it was amazing. This was a very motivated individual who was very excited about the biobank, and he just sent this to me one day in email, and I was completely floored. But we really wouldn't be able to do these things without the participation of the community. So the people are essential to make initiatives like this happen.
It is. It's just such an extraordinary opportunity for all of us, really. So I have this little screen here that says, "You are a presenter, show my screen," and I should click that, Kim?
Yes, click that. And the number of questions have come in that I think Liz will be able to answer, and we'll just hold those until after Suzanne's presentation, because Suzanne may get to some of the issues that people have asked about in her slides as well.
Okay.
And Suzanne, you're going to go to...
You guys see my slides?
Yep, but you're in slide sorter mode or something. You want to full screen it?
I'm trying to full screen it here. There we go. Is that full screen to everybody?
There you go. Yep. Looks good.
Okay, good. Okay, in, let's say about the next 15 to 20 minutes or so, I'm going to try to share with you why we believe the SolveCFS Bank is necessary, what makes the SolveCFS BioBank unique, and how the SolveCFS BioBank actually works. Kim and I were at a board meeting this weekend in Chicago, and it's really quite surprising, as I was telling the board, how many people really don't know what a biobank is. And that's just not for us everyday kind of folks. It's also for the professional community, the medical and the scientific community. So I think it's important that we all understand what our SolveCFS BioBank is and what it's actually going to mean to solving CFS. Okay, you guys have seen this slide many, many times, and I think it's an important starting point. I usually finish my presentations with this. I think it's an important starting point to give you an idea of really why the SolveCFS BioBank is a critical need right now. So let's just walk through this little schematic that I have that I think exemplifies a paradigm for CFS research. On the left-hand side of your screen, I have the world, and this is the world of CFS, which we know is a very complex, heterogeneous mix of people with CFS. Their CFS started at different times. Their symptoms are different. The triggers are varied. That result is an end picture of CFS, or a common pathway, common end final product of CFS that results in a very heterogeneous mixture of disease. What we're trying to do is ultimately get down to a very homogeneous, refined, targeted patient population that we can intervene. If we get very similar patient profiles or homogeneity, then we can really target interventions. So really, how do we get from what's very mixed to what's very homogeneous? The key is to identify biomarkers that can differentiate this complex mixture into something that is very homogeneous. And I think all of you would agree with me that the CFS community has spent years in pursuit of that. We have a number of academic institutions looking for indicators or biomarkers that help us get to this place. What we have had problems doing is really bridging the gap, essentially, between what goes on in academics and how that ultimately affects the patient at the bedside. So the biobank provides a much-needed resource to really bridge the gap between what we identify in the laboratory to what we can then bring to the bedside as far as treatments is concerned. So the SolveCFS BioBank is, in fact, a solution. It's an important solution that we haven't really had available to us for the duration of CFS research. It is a bench-to-bedside resource that will bridge the gap between what goes on in the laboratory and what ultimately helps the patient at the bedside. And I think by the time I'm done with my next few slides, I hope you'll agree with me. The SolveCFS BioBank is unique. It's very unique. I think what Liz showed, this framework, when I heard about this framework, when I was taking that public health genomics course, I was literally blown away. I could not believe that such an infrastructure existed for organizations such as ours, for patient communities such as ours, to be able to use to really help drive the direction that research was going in order to ultimately provide solutions. So the SolveCFS BioBank is very unique in the CFS community. It is completely customizable. We can customize it per the requirements of a particular study, and it's also scalable. We can do very small studies, or we can do very large studies. We can study not only CFS, but we can also, as Liz said, share our samples, share our clinical information with other disease groups, and also study other types of diseases. Again, we've talked before about how important it is to have control groups that are disease control groups, so something that's similar that helps us get clues about what's going on in CFS. The operational infrastructure of the SolveCFS BioBank is just a streamlined infrastructure that's really allowed us to launch the biobank in a matter of months. As Kim indicated, I learned about the Genetic Alliance BioBank infrastructure in September of 2009. We explored membership, we put together our application, and we joined in, I think it was earlier this year in 2010. And by March, our application was approved, and we were already hitting the ground running with our first study. That's because the Genetic Alliance BioBank has made institutional review available to members, and this is important because an institutional review board provides the necessary protection of people, that's human subjects, that are involved in research. We don't want anyone who wants to participate in research have any type of harm or adverse effect, so these types of steps are in place to help protect those who really do want to participate in research. The informed consent process or template was in place, and this helps groups like ours put together the information about what a biobank is, what participating in the biobank will entail, so that before you decide to participate in the biobank, you understand what you're getting into, and helps you make it when you sign on the line, you are agreeing that you understand what you're into. And for us, for the SolveCFS BioBank, it's an opportunity to participate in all different types of research. The SolveCFS BioBank, or the Genetic Alliance infrastructure, provides a web-based system that allows us to collect extensive and comprehensive clinical information about you. And this information is linked, it's attached to that biological sample that you provide. It also provides the standards for collecting the sample, for processing the sample, and tracking and storing the sample. So even though you may be in Colorado, your sample sits in Maryland, and it might get distributed to Canada for research, all of that is maintained in a very integrated information and tracking system, along with being able to manage the data effectively and providing tools in order to be able to query this biobank, which again is a database of not only clinical information but biological samples. So we can, let's say, for example, we have an investigator who wants to look for a particular stain in blood, and they only want to study people who are 20 years old and that are white and have been sick for 2 years. We can type that information into our data management system and pull it out and see who in our biobank meets that criteria. So it really is an incredible bank of information, just like a financial bank, you know, where you have an account set up and you deposit your money and you take out your money, and all the other services that the bank offers because they know something about you and something about your needs. This is very similar, except now we're dealing with biological information, both on a clinical level and at an actual biological sample level. And as Liz indicated, the infrastructure has very strong security and access control built into it, so not anybody can get to this. It's very strict on who we share the information with, which is investigators that apply to us, and then they always get de-identified information. So again, just an unbelievable resource that ultimately will serve as an invaluable knowledge base that not only has the expertise that we bring to making the biobank a resource for the research community, but the knowledge and expertise that you as a patient and a possible participant bring to the biobank. The SolveCFS BioBank works because it's an infrastructure that's been standardized. And we have made, I think, the very important decision to collect the same standardized information using questionnaires and questions that have been standardized and validated for a number of different studies, for international studies, and for that the CFS community, research community, and medical community have been using for years. This way, ultimately, what we can do is we can extrapolate or generalize the findings from our study, not only to other CFS populations, but potentially to the bigger populations at large. For example, many of the questions that and the questionnaires that we use are also questions that are asked and used in the NHANES survey. That's the National Health and Nutrition Evaluation Survey, which is a study that's been going on for many, many, many years. And so the information that we collect can also be generalized or put in the context of NHANES. So when you receive your forms or you log into the web-based system, the information that you will be filling out is general demographics, that's your age, weight, height, sex, where you live, how long you've lived there, etc. General health questionnaire, which is very much like the SF-36. It's a wellness and disability questionnaire that assesses things from physical activity and physical function. Also the multidimensional fatigue inventory, or the MFI, to assess the levels of physical and mental fatigue that the participant is experiencing. We're also using a symptom inventory, and this is a much more comprehensive symptom inventory than what has been used previously, because now it includes criteria from the Canadian case definition. And this was made possible by Lenny Jason at DePaul, who has helped us make this particular instrument hopefully a little bit more specific for people with CFS. You will also fill out a brief pain inventory questionnaire, a sleep questionnaire, a very comprehensive medical history survey, and a family history survey, so that we can try to figure out what else is going on, or some of the risk factors that are involved in CFS. And finally, a medication checklist. So it's a very comprehensive workup that, set of questions that you will answer in order to populate that data that goes into the biobank that ultimately links with your samples. And eventually, we hope to be able to merge the biobank information with the patient's actual medical record. And that has not been done yet, but these are things that we're looking towards in the future. What about the biologicals? Okay. As a researcher, it's hard to emphasize further or more the importance of the sample. The biological sample that goes into any assay is unbelievably important. And what I mean by that is the type of sample and also the quality of the sample. This particular tube pictured over here is actually a separated blood sample. And I just showed this to you because I wanted to be sure that you understood that blood is a multi-component sample. It's not just a liquid sample with red blood cells. It's also a liquid sample free of red blood cells that has many other types of molecules that are important to measure in it, such as cytokines. And there's also a cellular portion to the sample, which in this case contains white blood cells and platelets, platelets being the cells that produce antibodies. So it's a multi-component sample that is very, very precious. In CFS research, we've used it really as a sentinel sample. A sentinel sample in the fact that the blood travels throughout the body and can give us some clues to perhaps what's going on elsewhere, whether it be in the gut or in the brain, and also a sample that may in fact provide us clues to the ultimate cause of CFS. So I can't emphasize more how precious this sample is and how important it is to make sure that the quality of this sample is as best as it can be in order to be able to analyze it to get at those things that we need to in order to solve CFS. On the picture at the bottom shows not all of the different types of tubes that blood can be collected. And actually, this is just a subset. There's probably about another 5 or 6 more different types of tubes. These collect serum. That means blood gets drawn into here, and the blood clots at the bottom, and the only thing that's left up here is a fluid that is an acellular fluid. That's called serum. The tubes that we're collecting right now for the particular ongoing study is this green top tube. And the reason I'm showing you this is that each one of these has a particular way that they need to be handled. So for those of you participating in the current study where we get green top tubes, it says make sure the phlebotomist inserts the tube 6 to 8 times and then puts it into the collection kit, or the kit that sends it back to the processing facility. So, I mean, that's quite specific. There's other types of tubes that have even more specific types of instructions. So when you participate and when you get your package in the mail, be sure to read the instructions so that you know what will be, what needs to be done to your sample by the phlebotomist in order to make it the ultimately best sample that it can be before it gets into the freezer and into somebody's assay in the laboratory. So collection and handling is so very, very important. Also, the processing and storage of that sample is very important. And again, the way that the infrastructure has been built for the SolveCFS, or set up by the Genetic Alliance for the SolveCFS BioBank, means that we can track the labeling of these samples is standardized, the tracking is standardized, so that we know what is in our inventory when investigators apply to the biobank to conduct their research. Another sample that we're collecting is the Buccal swab, and this is just basically kind of a rough sponge on the end that you stick into your mouth and you scrape on the side of your cheek in order to isolate cells. And these cells are then sent to the biorepository in Maryland, and they isolate DNA. This DNA can be used for genetic studies, and it can also be used for a number of different types of things, such as cataloging the types of DNA in the mouth. Remember, you guys know that I'm one who likes to think a lot about our metagenome, or all the different types of genomes that are hanging out in our bodies. So this would also be another good use for this type of sample. And it's a very stable sample. So it's not as susceptible or vulnerable to the types of collection and processing events that the blood is, but nonetheless, it's an important sample that should be taken care of. So when you get it in the little mailer, you take your cheek swab, you put it back in the mailer, and you send it so that it can be processed and stored appropriately. And again, all those instructions are in the kit that you would receive when you participate. So we're really believing, we see that the SolveCFS BioBank is a catalyst for translational research. It is what has been lacking for so many of the studies that have been going on throughout the country, throughout the world for that matter, that have found some very promising biomarkers but haven't necessarily had access to the types of samples that are needed in order to be able to validate those findings and then bring them to the next level, or replicate the findings of another investigator. So I think you can see that this is a very important, a critically needed resource in order to advance CFS research. And we already have tons of opportunities for research, and it turns out to be a very effective cooperative model for research, where we're engaging all stakeholders, whether it be physicians, researchers, patients, advocates, pharma, everyone that applies and whose application is accepted, or anyone who consents to participate. This really does translate into a very cooperative model for research. And the biobank itself will serve as a hub for some of the highest quality biological samples and standardized clinical information available for CFS. So finally, we need you. This is a new announcement. We are now opening the SolveCFS BioBank for general enrollment. When we're finished with this webinar, you can go to the SolveCFS website and check out what the criteria are. But we've opened it up quite a bit, as you'll see once you get to the website and look at the criteria. And I think that probably all of you listening today will likely be eligible. And I think one of the reasons that we're doing this is because the way we've actually decided to do it, I should back up a little bit, is we are going to open enrollment, and what you will do when you decide whether or not you want to participate by checking out the criteria is you'll contact the biobank coordinator, that's Gloria, and Gloria will put you through the consent process or share the consent process with you. And when you consent to participate, you will provide all the clinical information that I talked about a few slides back. That's all the questionnaires. You'll fill all that information out. And then we'll have that in the database. We will not collect a sample from you until there is an investigator that needs a certain, that is doing a specific study that needs a certain type of sample. This again speaks to how customizable the biobank is. We can collect, whether it be one of those red top tubes or a yellow top tube, we can customize it to the specific needs of that investigator. And you can see how that would be very expedited because you as a participant would already be consented to as a biobank participant. And if you meet the criteria for that particular investigator, then Gloria requests a kit to be sent to you. You get the kit, you get your sample drawn, and then it goes right into the biobank. So what would have before taken months to a year to get started will now take literally days, a week. So this is why we've made the decision, and I think it's a very important one, to open up the SolveCFS BioBank for general enrollment. So please go to the website and check out the criteria. Now, we not only need you, we need your family, and we need your friends. We need to build this biobank so that it is a resource for us to figure out CFS. The only way we're going to figure out CFS is if we can compare people who are sick to what's going on in healthy family members and what's going on in healthy friends. I put the example down here of the Susan B. Love Foundation. Susan B. Love Foundation has an initiative called the Army of Women. And what the Love Foundation has been able to do is enlist women from all around the world to agree to provide various types of clinical information and biological samples in order to solve breast cancer. So these are women who may know someone who has breast cancer or may not, but they've volunteered because they are committed to the cause and they are part of this army of women who are going to solve breast cancer. We can do the same thing for CFS. Together, we can solve CFS. Now, I haven't thought of a really cool name like Army of Women. Maybe we can all be the conquerors of CFS. But if you have some good ideas of what we can call ourselves, our family, and friends who want to really be a part of the solution, who really want to be part of this initiative, we'd be really happy to take your considerations for what to call ourselves. But for now, it's Conquerors of CFS. But please, when you participate, think about your family and getting them involved, and think about your friends, because we not only need you, we need your family and friends too. And I think that's it for me. Is anybody out there?
Yes. Sorry, I forgot I was muted. Thank you, Suzanne. I'm sitting here busily answering questions, and I fear that I have not been able to pay as full attention to you and Liz as I wished I had, but we're getting just great questions and lots of interest and participation, and that's what our primary objective today was. So,
Woohoo!
So, Liz, you're still with us, right?
Yes,
Are you
I'm still here.
You're unmuted, and you sound a little softer than Suzanne does. So if folks...
I'm bouncing on my ball and I'm shouting. I'm so excited about this darn thing.
One of the first questions that came in during your presentation, Liz, was if you could just address both the security provisions that Genetic Alliance has made part of its infrastructure, and one other person asked about GINA, the Genetic Information Non-Discrimination Act, and since Genetic Alliance played such a huge role in getting that legislation passed, if you could just touch briefly on how that protects people who participate in the Genetic Alliance biobanking infrastructure, which includes the SolveCFS BioBank.
Okay, so I will start with the first one about the technology and the security aspects. So I want to direct you to a resource, the BioBank website. It's biobank.org. There's a tab on the left-hand side that's a frequently asked questions, and that goes into a lot of the detail. And in general, with technology, with the security aspects of it, we have, there are multiple levels of users and access through passwords. All of the groups have administrators of the biobank, and they can determine who has which levels of access into which systems. We also have firewalls between all of the individual groups, biobanks, and registries, so all of that information is separate and it isn't shared across. Now, the groups could decide that they wanted to collaborate on something and they wanted to share data, and if that were the case, then they could share the data, but it's at the group's discretion. And we also have audit trails that capture all access. One of the great things about going with a biorepository vendor that does this for a living, we're really just a very small piece of everything they do, and they have many, many pharmaceutical customers and people in industry, and they require a level of security and access that is needed so this data can be compliant for FDA submissions. And we're just really fortunate that they have to meet this rigor, and we can take advantage of it as well. And then they have multiple backup systems on all of their equipment. They have this really ginormous on-site liquid and nitrogen tank that will hold things for a very long time, and they've really done a nice job, and we feel very secure with our data and samples that are there. And we know that these are really, really precious resources. And there's more information on the FAQs in terms of specifics that I might have forgotten to mention. So that was the first question. The second question is about GINA. We are developing some information for consumers about GINA and what the legislation means to all of us. And those are not yet available, but when they are, I will let Suzanne and Kim know. The thing that's really interesting about GINA is it really only protects people that don't already have, if you have a genetic condition and you are already diagnosed, GINA doesn't protect your health information from becoming available because it's already known that you have that condition. So I think that we're going to, it's going to take a while to really understand how this affects all of us. And the rules, the way it affects us will be different for those who've actually been diagnosed with a genetic condition and those who might just predispose to it.
Sorry. Thanks, Liz. I'm sorry. I'm trying to do 4 things at once now, which I guess is fewer than usual. But that was great answers, particularly on the security question, which a couple more people have written in about. And also, kind of a related question is a concern that insurance companies or employers, that other people you know, might use information captured by the biobank against the individual, and maybe one of the two of you can just reiterate how restricted access to both the sample information, the collected information about the individuals, and also the samples is.
So let me take that one first, and then Suzanne, you can jump in. So only certain people have access to the records, and we know who has access and when they're accessing it through the audit trail. When a member organization gives out data and information to researchers, so when the SolveCFS BioBank gives samples and data to researchers, all of those samples are de-identified. So that means that there are no identifying information is transmitted with the samples. And so that's really a way to protect you. Now, the people at SolveCFS BioBank, they do know who the samples came from, and that's really important because then they can always go back to you. But the researchers are only getting de-identified information, and the researcher will not be able to identify you. And I mean, and that said, there is some discussion right now in the rare disease community with some of these diseases where there are maybe there are only 30 kids in the world that have a condition. In those situations, it's very, very hard to de-identify because the people in the field already know who the 30 people are that have the condition. So when the paper comes out and it says that there are 2 children in Europe, they can usually figure out who they are. So that's a rare example where it's hard to de-identify. But we do everything we can to protect your information. Suzanne?
No, I think that's perfect. Absolutely perfect explanation.
And this question's actually for either one of you. A few people have asked if something is discovered using samples from the biobank, does the individual participant get notified about that discovery with regard to their own sample?
No, they do not. The individual does not get notified, whether it be a particular result or a particular study. Now, the only way that that's a little bit different is if, for example, a third party, one of the investigators that we share samples with on an approved study, if they make a discovery and they then need to work with that particular individual, they then come back to us and we contact that individual to determine whether or not they want to be contacted by that third party. And if yes, then that third party can contact the individual. If no, then the third party cannot. Does that make sense?
Yeah, and can I add a couple of things to that? One of the things that the SolveCFS BioBank is going to require is that the people that are using their samples are reporting back their results. That's in one of our approved template documents. So you will get updates through SolveCFS BioBank communications, and they'll be general, and they won't be specific to an individual, but you will be knowledgeable about what's happening with the collection. The other thing is, let's say an investigator learns something that's really important to the community, then the biobank will contact everyone and say, we just found some information and we think you need to go to your doctor to discuss further. Because one thing you have to remember is all of these samples are for research, and research and clinical care in the United States are very separated. And the kinds of assays that happen for clinical diagnosis, for genetic testing versus for research testing to see which genes are involved, they're completely separate processes. But it doesn't mean that if they find something, you'll never get the information. You might get it in some form and then need to go follow up through a different channel, because we just have to remember these are research samples, and we can never use them in the way that we would clinical samples for, not clinical samples, but for diagnosis.
That's right. It's research. It's not standard of care. So the only way an individual will find out a result is if that third-party scenario happens, like I just explained. So, for example, those of you who might be interested in a virus or XMRV, and if we did a study on XMRV, individuals would not receive those results, but we would report them out in general. And all investigators that use samples from the biobank are required to publish their results, whether they are positive or negative.
Here's a question kind of along those lines about the sample. A couple people have asked whether freezing the sample destroys any important information that might be in the blood, and whether we foresee being able to collect and make use of materials other than blood and buccal tissue.
That's an absolutely great question. Yes, freezing blood or some of the blood components can affect the quality of it. That's why it's so important for investigators when they design the study to design the collection of that sample in a way that's appropriate or conducive for the particular assay that they're using. So we are collecting samples right now that the cells are, that one little layer of white blood cells and platelets that I showed you are collected and processed in a way that the cells are viable when they're unfrozen. So that's a very specific, particular way of processing cells. And then you can do a number of things. So yes, freezing does affect the quality of the sample, but there's also very specific ways of freezing and treating the sample so that it gives you the type of molecules and information that are the correct result. But that's an excellent question. The sample processing is so very important. You have to know how to handle and process that sample in order to get the right result.
And that's actually, to me, one of the unique features of the SolveCFS BioBank is the fact that we have the opportunity through the Genetic Alliance's infrastructure for ethical recontact. So if a researcher comes to us and gets a study approved through the many hoops they have to jump through for us, and it requires a different sample than what we have in the biobank, we can easily go back to all the people who are enrolled and consented to collect those samples.
Exactly. Yep. Good point. And no, it's not just limited to buccal swabs and blood. Again, we can collect collect other types of samples, whether they be tissue samples or urine samples. It's really a customizable biobank and biorepository.
Yeah, so for now it's more a resource issue of what we have the capacity to do due to really financial resources and the support of that.
That's right. That's right.
A couple people have tried already to access the criteria on the website, and I just wanted to let everybody on the call know that we're checking that out on our end, and hopefully we'll have that little computer glitch resolved. A couple people who are joining us today from outside the USS, particularly Canada, wanted to ask about participation in the biobanks. Suzanne?
We are the bank without borders.
So,
right, Liz? I mean, Liz, you might be able to address any of the issues that you've run into with some of the other members that, for the rare disorders where they are trying to find people throughout the world with a rare disorder. But as far as I can tell, you know, where FedEx goes, we go.
Yeah, absolutely. And we use some different international carriers. We just had a group that did a very large family collection in Libya. And we just used a different kind of tube called a Paxgene tube because they tend to be a little more stable over a longer period of time. If you are international, it's totally feasible. We usually just work a little closer with the international donors because there's a little more logistics around the samples because it has to go such long distances, and you definitely would want to make sure you would ship it earlier in the week. And even as part in the U.S., you know, we really recommend ship Monday, Tuesday, or Wednesday. You know, it's going overnight, but just in case. And you know, if you're in Europe, we really want you to ship it on Monday just because we want it to get there as soon as it can. So absolutely everyone can participate, and that's just another great thing about this system is you can be anywhere and participate. And in many research studies, they're often run at centers, and maybe, you know, in Denver there is a center, but if you live in Chicago and there isn't anyone doing that study, you can't participate. I just picked those randomly. But the nice thing here is no matter where you live, you can participate.
So somebody has asked about the cost of being a participant in the biobank, and at the present time, the association was able to undertake this really innovative endeavor through the support of our general donor base. So all those who have supported the organization have helped make this biobank a reality. The only cost to individual participants in the biobank is that we don't have a way right now to offset the cost of you having your blood drawn. The kit and everything that's needed to ship the kit, the shipping costs are all covered, but occasionally the phlebotomist, the doctor's office will have a small charge, usually between $10 and $40, just for the process of drawing the blood, which they will sometimes waive if they understand that this is for research. And the kit that you receive includes instructions for the phlebotomist and reinforces the fact that this is for research purposes, and hopefully you know, you can work with your primary care doctor or the lab that you're taking your kit to get that cost as low as possible. Suzanne, did I miss anything on that? Or Liz, you might have some other.
Nope, that's perfect.
Okay.
Yeah, I just wanted to say, there's by and large, phlebotomists very often waive the fee. They think this is so cool that people are participating in research, and there are a lot of creative ways to get your blood drawn at lower cost. And some of the groups have, you know, gone to their school nurse. You might have a friend who is a phlebotomist or an EMT, but like I said, most clinical labs, they just think it's so cool that people are doing this. We had one example where someone had to charge for the needle because they actually inventoried the needle, but I think that was like $2. But yeah, by and large, it's usually covered. Or free, I guess the word I mean.
Yes. And that's just really exciting. I mean, there aren't very many other ways in which individuals can participate directly at such minimal cost to themselves. Most studies require some sort of travel, and it's just really cool. This question, I think, is probably more appropriate to Suzanne. Somebody asked about at the time of that the blood is collected, how do we account for the specific sort of clinical situation of the patient, whether they're in a relapse or, should they exert themselves immediately before they have the blood drawn, or should they, try to rest up for a few days? Any general guidelines, and how do we accommodate that in understanding what their results might be?
That's a great question, and right now we don't have any of that built into the sample collection, and I think that's very, very important. So whoever that was, thank you very much. We will build that somehow into our kit, because you're right. If you have to drive to a lab to get your blood drawn, and maybe there's some steps or something to walk up into the clinic, that is a stressor that we're not necessarily accounting for. So we'll get on it. Right now we don't have any controls, but that's very, very important.
I'm not really surprised.
No, that's a great question.
Yeah, it is a great question, and the use of an exercise paradigm for CFS research has become very popular following the publications from the University Pacific group that showed that things are more pronounced after patients exercise than as compared to when they are in a rested state. But this isn't really set up to test patients under that sort of research exercise paradigm. And that would be a hard thing to enforce consistently across individual circumstances of having blood drawn, but certainly an important thing for us to take note of.
Yeah, although we could in fact design an exercise study and use the biobank for the collection of samples, but that would have to be built into the design. This is the point that person raised is so very important, and we're actually having to respond to the journal Brain Behavior and Immunity regarding the paper that we just published on cytokines in people with CFS, because there are a couple of investigators from around the world who asked, well, how did you control for how well that person slept the night before? And how did you control for the types of experiences that person had before their blood was drawn? And those investigators are asking the same question that our participant just asked. It's very, very important information. So thank you again for that.
The other question, I think this one will be our last since we're already over time, is similarly, how do we control for different medications that individuals are taking?
That's sometimes study specific, so certain studies will require that an individual can't be on a certain type of medication or any medications at all. This is one of the reasons why we've made the decision to open up enrollment for the biobank, because we will get a much larger selection of patients and people to select from for specific studies. So that's why we collect that information. So there's a very extensive medication use checklist that people will fill out, and it collects information on practically every type of medication that affects the various systems, from the central nervous system to the cardiovascular system to sleep, etc. So if an investigator requires medication-free patients, hopefully we have those in our biobank. But again, that goes to the power of this biobank infrastructure and database because we query it just like going to the library and finding, finding the proper book or the proper recipe or the proper resource to fit the needs of the investigator.
Yeah, great questions, and although we're out of time, I want everybody to, who's still on the line, which is almost everybody who started out with us, it's been great retention during this program, to know that we have a log of all the questions that were asked during the webinar as well as all the questions that came in ahead of time, and we'll use those both to help shape the evolution of the biobank and also to address questions that are coming up frequently in materials that we post on our website and in our monthly e-newsletter SolveCFS. I'm sorry, CFIDS link, getting all my names mixed up. Take a second here to just point out a couple of webinars that are coming up. As many of you know, we have this stable core of people who have come to every webinar that we've put on so far, and we just love our frequent flyers who become familiar with the technology and are able to use it and get past sometimes the little glitches that occur. So thanks for doing that. We have 3 coming up in the next few weeks. Suzanne is going to wrap up part 3 of a 3-part mini-series about the association's research program, and I apologize for the typo there. That'll be on Thursday, June 17th. And then we have a little break because of the holiday. But on Thursday, July 15th, Dr. Vincent Rocchianello and Dr. Cindy Bateman will do kind of a team approach to providing an XMRV update. And many people are familiar with Cindy Bateman. She is an expert CFS physician, clinician who's dedicated her practice to taking care of people with CFS and fibromyalgia. She's also involved currently in a study of XMRV taking place at University of Utah, and she is the clinical coordinator or the clinical partner to the University of Utah study that the association is funding with Kathy and Alan Light. If you don't know Vincent Rocchianello's name, that's not unusual, but he's perhaps better known as the TWIV guy. He has a weekly podcast called This Week in Virology and has covered CFS and XMRV since the first paper came out in October, and just real excited about his willingness to join us for this webinar and talk about XMRV, the agent, and kind of more broadly how research connecting an infectious agent and a condition usually proceeds in the world of science, and then Cindy can focus more specifically on CFS and the ongoing studies. And then this is another announcement just as of today. Dr. Louis Katz, who is one of the world experts on blood safety issues and participates as a member of the Interorganizational XMRV Task Force that's been convened by the American Association of Blood Banking, on which I also serve, accepted our invitation this morning to do a webinar on August 12th on XMRV and blood safety. And Dr. Katz is at the Mississippi Valley Regional Blood Center and again is a renowned expert in transfusion medicine. So in addition to our own cherished experts, we're really delighted to involve these folks who may be new to CFS but are world experts in their own right. And you can find the whole schedule going all the way through December on this link at the bottom of the screen, and it will also be included in the follow-up email message that we send to you in the next day or two once we get everything posted up from today's program. We'll let you know where that is and also where you can find these other resources and get registered for the next set of webinars. So I think that that concludes our program for today. We've still got almost the entire audience with us, so thanks to everybody for hanging in there and making use of this cool but somewhat foreign technology. And thanks so much to Liz Horn and Suzanne Vernon and for all of our SolveCFS BioBank participants, current and future. Now that we've opened it up, we anticipate that Gloria and Lauren are going to be very busy enrolling new folks, and that's just really exciting. And our board, having met in Chicago this past weekend, made that decision, and we couldn't be happier. So the last message I will leave you with is that things and developments and activities are occurring at an unprecedented rate right now. I have been with the association for almost 20 years, and there's been no time like right now where things are changing as rapidly as they are. So please stay connected, remain hopeful. Change and progress is definitely on its way, and we hope you'll follow us through CFIDS link and our website, and as supporters of the association, and stay in touch, and look forward to sharing new announcements with you regularly over the coming weeks. So bid everyone farewell and a good afternoon, and thanks for your energy during today's program.
Thank you. Bye, everybody.
Goodbye.
Bye.
Bye, Liz.
Thank you.